So that this proton by itself is what changes the pH of your blood. So if your pH drops too much, then these protons are going to be more likely to come in contact with the bicarb because there's more of them and be absorbed to become carbonic acid again. So remember, can you guys hear me? Somebody can't hear. Okay, there's a few questions. I'm going to stop for questions. Sorry. Yeah, so it's just so how is it that the lower the pOH, the more basic? Wouldn't it be the higher the pOH, the more basic? So yes, it is kind of confusing in that sense, but really you're just finding the negative log of the concentration. So if the concentration of OH or hydroxide is high, that means it's more basic, right? But when you find the negative log of that, it's going to be a lower number. It's confusing because it's like the opposite of what you'd think. That's like why it's confusing. Oh, somebody answered that. Great. Okay. pH of 8 is less basic than pH of 10. pH of 8 is more acidic than the pH of 10. So 10 is more basic than pH of 8. Does that make sense? Hopefully I answered your question. Okay, moving on. So, if you have a lot of carbon dioxide, so say your body's retaining more carbon dioxide for whatever reason because you're not breathing properly, then the reaction is going to shift to the right and make more protons, right? And so these are going to decrease the pH of your blood, which is bad. However, say you just finished doing a strenuous workout and you are releasing a lot of lactic acid into the blood. Now your proton concentration is going to increase. So in order for your buffer to kind of reduce the amount of protons, now your reaction is going to move to the left so that it can absorb it and turn into carbonic acid and reduce the amount of protons floating around. Does that make sense? Hopefully that makes sense. Okay, so I kind of drew out the phosphate buffer just so you guys can see it, but it's low yield, really. Just know that it exists, but it's not as important as the other buffer, the carbonic acid buffer. The protein buffer, I noticed somebody, I think yesterday's lecture had a question about this in relation to like hemoglobin. So I'm going to try to like describe this in the best way possible. So when your blood reaches your lungs, the oxygen concentration is going to be high, right? Because you're breathing in all this oxygen. So the oxygen is going to bind to the hemoglobin, specifically the iron group. So you don't have to worry about this too much because we're going to go over it when we learn about hemoglobin in a few lectures. So this is just kind of like an introduction. All right. So as that blood travels to, say, your leg muscles, where, you know, your leg muscles are secreting a lot of byproducts like carbon dioxide from metabolism, you're going to have a higher amount of carbon dioxide in your leg muscles. And so because you have an increase in carbon dioxide, you're going to have an increase in protons. And that's going to drop your pH in the tissues. So this is kind of where you'll run into your protons, which will bind to the protein aspect of the hemoglobin, so the globin. And this will knock off the oxygen. So this is pretty much how oxygen gets to your tissues, how it knows to get off the hemoglobin so that it can enter your tissues. Okay. So for these, know the 3 different maintenance techniques in your body that keep your pH normal. So your acid-base buffers, we talked about those. Those work instantly. Then your respiratory mechanism takes a little bit longer, but this is where you increase your respiration to blow out a bunch of carbon dioxide so your pH can go back to normal. And then your renal mechanism, this is where you pee out a bunch of bicarbonate to kind of also balance out your pH. But don't worry about that, that's more physio. I kind of gave you guys an idea here. So if your pH increases, then your kidneys are going to dump out a lot of bicarbonate, and that's going to allow your pH to drop. Remember, bicarb is a base, so when it when it, decreases, it's going to drop your pH to a more acidic value, and then you go back to normal. Alternatively, in your lungs, if your pH drops for whatever reason, your lungs are going to blow out a bunch of carbon dioxide. So your carbon dioxide in your body is going to decrease and your pH is going to increase. So you'll go to a normal blood pH. Okay, so if everybody can participate with this question, just
can you
type
go
your
back
answer
real quick?
Yeah. What? This slide? Okay, so I've kind of thrown in some practice questions in this PowerPoint, so just type your answers in the chat. Are you good with this slide, by the way?
Yes, I'm good. Thank you.
You're welcome. All right, yeah, just type your answers in the chat. Okay, awesome. It looks like everybody's choosing B, and that is the correct answer, because remember, if your protons increase, you're going to shift to the opposite direction, which is the left. Great. Did anyone not understand that?
Yeah, I have a question about this. So what do you mean by it's shifting to the left? Do you mean it's shifting to the left to produce carbon dioxide? Or I just don't understand what you mean by that.
Okay, sorry. Yeah, it is kind of confusing. So pretty much your body's going to want to, H2CO3. Sorry. It's going to want to make a proton and a bicarb, right? So if you have an increase in protons, they're going to be more likely to react with these. So just if you have more protons, they're going to be bouncing around in your blood and they're going to react with these. So then they'll go towards the left and make more of the carbonic acid, which is pretty much your buffer system.
So you're saying I'm confused. That's shifting to the right though. According to the you look at the equation on the other slide you had,
Oh, this is separate. This is more of what I'm talking about, your buffer system, because carbon dioxide, which is a byproduct of metabolism, it reacts with the water in your body to make this, right? So I'm referring to this half of it, because this is what you're going to see more often in your body is just the protons with the bicarb, and it's reacting with each other to make carbonic acid to kind of buffer it. So this is kind of just saying how this is made in your body, right? So essentially you would, if you did, you would have to go all the way backwards, but it's all, it's the left regardless.
Okay, so is the carbon dioxide and water reaction, is that just that's what happens physiologically to carry it, right? And then it turns into the H2CO3.
Right, yes.
To transport it, okay.
Yes, and this is the main buffer system, right? So it together, this is what carries the proton. Does that make sense?
Yes, I get it. I just didn't know. I was confused about what it was being transported as, and I just didn't understand what you meant left relatively to.
Yeah, no, it's okay. It's kind of confusing.
Thank you.
You're welcome. Okay.
Phoebe, do we get a copy of those questions or no?
So I'm kind of on the fence about that because I want you guys to, participate and show up for those. So I was thinking of not making those, but since you guys aren't getting the full amount of questions, I might just post them as well, if you guys would prefer that, of course.
Yes, if you could post them after the session, that would be great.
Yeah, okay, great. I'll do that then. Okay, if nobody has any more questions. So here is your Henderson-Hasselbalch equation. You're going to have to know this for sure because there's definitely going to be a question on the test about it. That is very similar to the example she's provided here. So you'll see that she used a log of 1, so 0.5 over 0.5. She's going to give you something like that. Does somebody have a question? I see like a hand raised, but oh well. Okay, so she's going to give you something that is equal to 1. I've also kind of included some logarithm, what they equal to, just so you guys can memorize that. But pretty much if you're finding like the log of x, for example, if you're trying to solve for that and say it's 2, you're going to make it base 10. So 10 to the power of log cancels out. You do that to the other side, and so x would be equal to 100. Right? So that's pretty much how you would solve for that if she asked you. But I did, include it right here. So memorize this formula. Okay, so with this, she's not going to give you like something like this, but it's good to understand why pK is equal to pH, and that's what makes it the best buffering range, right? So if you have an equal amount of conjugate acid and an equal amount of conjugate base, you're in the middle, right? So you're able to move in either direction more easily to buffer. So this is kind of the idea here. This is the 1-to-1 ratio that where pK equals pH. And so you can move in either direction more easily. If you want to like look at the math of it, if you just plug this into the Henderson-Hasselbalch equation, you can pretty much figure out that pH is equal to pK when acid is equal to base, or the ratio is 1-to-1, which is pretty much just saying that base is equal to acid. So another thing to note about this is that if you had a buffer that has like this kind of sloped line, you see that with a smaller change in the ratios, you're going to have a larger change in the pH, right? So that's pretty much indicating that a buffer in this red line would be a weaker buffer than this other red line, right? Because a steeper slope will indicate a smaller change in the pH with a larger change in the ratio of acid to base. So keep this in mind as well. She may ask about it. Okay, so this is more physio than biochem, but it is important to kind of understand the difference between acidosis and alkalosis, as well as metabolic and respiratory of each type. So acidosis is where your blood drops below this pH. So your normal physiological range, is 7.35 to 7.45. But we use the middle number, 7.4, when we're doing the math. So if you have like an excess in ketone bodies or a loss of bicarb where your blood pH is going to drop, you're going to know that as metabolic acidosis. So if it's something where your body is making it happen. If your lungs are doing it by retaining CO2 in certain respiratory illnesses, you're going to have an increase in CO2. And remember that if CO2 increases, you're going to have an increase in protons. Alternatively, with alkalosis, if you have a drop in CO2, you're going to have a drop in protons, and you're going to wind up with alkalosis. And with metabolic, you're going to retain bicarb. So this is if you like just eat a bunch of baking soda and your body metabolizes that, and then you have too much like base, and so it's, you're going to increase in pH. Hopefully that makes sense to everybody. You don't have to know these little details because this is definitely physiology and you're not there yet, but just kind of have an idea the difference between metabolic and respiratory for each of these. Okay, another question. Okay, awesome. Looks like everybody is saying E, and that is the correct answer. So don't make the mistake of forgetting this formula or getting the things confused with one another, because that is definitely going to be a question on the exam. And if you have the wrong formula memorized, you're going to get the answer wrong, and that's going to be sad. So memorize this formula. All right, so we're going to start with the amino acid. For this, literally just know asparagine was first. Don't remember the date. Don't remember the names. That's too much information. This isn't a history course, so don't worry about that. For this, kind of know Miller and Urey, and that they were able to make amino acids using this and an electric spark, which is supposed to represent lightning. But it's low yield, so don't worry too much about it. Okay, so we know that the nucleus has the DNA, and the DNA is transcribed. The DNA is transcribed into RNA, and then from RNA to protein. This is genetics, don't worry too much about it, but it's just going to tell you that these are going to be RNA codons, so not DNA codons. Don't make that mistake. Obviously, by the U, you can tell that they're RNA codons. Don't remember any of these except for the ones I've highlighted, which are the start codons and the stop codons. I don't know if you guys have fun mnemonics for these, but I'll share a couple of mine. School starts in August, so that's a start codon, and it codes for methionine. So know that methionine will start every protein sequence. And then for the stop codons, I have like somewhat of a phrase to remember those by. So you are annoying, you go away, and you are gone. I think I stole that from the Princeton Review, but if it helps you guys, you can use it. This is a transition slide, don't worry about it. This is just an introduction. We're literally going to learn about all of these examples later on as we get through the course. So she's just kind of introducing the idea that, you know, maintenance, energy, and regulatory are all things that proteins play a part of. Okay, from here, know that the peptide bond is a covalent bond, that there's 20 amino acids that make up proteins, and that you can enzymatically modify the amino acids. So this is going to be important when we go over collagen formation, because you'll see that the amino acids will have like a hydroxyl group or a sugar group added to them, and to kind of change them a little bit. And that's important for the formation of collagen, which we'll go over later. So just have an idea that that exists. So know that the codons are the 3 nucleotides that make up the code for the amino acid. And then the genetic code is also kind of important to remember, but this is just basic stuff that you guys already know about. And know that there's 64 codons, but that's all low yield stuff that you're assumed to already know. So for all this, I kind of gave you guys an example of what we're going to go over. Just know that amino acids play a role in all of these. We're going to go over pretty much all of these later on in the course, so you don't have to worry too much about it. It's just kind of like a transition slide. Okay, know this forwards and backwards. You know that there's an amino group, there's a carboxyl group, and that the middle carbon is the alpha carbon. Oh, there's a question. Is this PowerPoint going to be on Canvas? Yes, I'm going to post it later today. Okay, so yeah, know that the alpha carbon is the middle carbon that is also connected to the side chain. So I've kind of drawn out the reaction that happens, but you don't have to know the reaction. It just know that water is released from this reaction to make a dipeptide, a tripeptide, and ultimately a polypeptide. Okay, for the amino acids, like I said, I personally didn't remember the shape, but she said to, and you probably should anyways for the MCAT, but they're not going to ask you to draw it out. So having a general idea as to what they look like will be useful, and should also be helpful for categorizing the different types of side chains. So memorize these, know the name, know kind of the basic idea of what the side chain is, and definitely know the 3-letter and 1-letter abbreviations, because on the test there was a question that only used the 1-letter abbreviations for amino acids, and we're supposed to know what those meant. So I'm kind of just going to breeze through these because you guys just have to memorize this and have an idea as to what they are. I've added this kind of like summary slide for all 4 of those slides, so if it's useful, you can use it. If not, don't worry about it. All right, another practice question. This is more for those people who've already started memorizing these, but it's an easy question, so. Great. Most people are saying C, which is the correct answer. Great job. Okay, so for the rare amino acids, don't worry about the structures. That's too much information. And honestly, if she does ask about one of these, it's going to be about selenocysteine because that's the one that's found in mammals. And this is, a medical course, so it's going to be more about mammals than it is about a bacteria, right? So, I mean, just kind of have a general idea that these are stop codons that, you know, code for them. But honestly, this is not going to be tested as much or as in depth. Maybe they'll mention selenocysteine as like one of the possible answer choices, which is like a wrong answer choice. But kind of have an idea as to what these are and, the stop codon sequences and maybe know selenocysteine to a little bit more depth. Okay, like I said, 3-letter, 1-letter, and amino acid, know all this whole chart pretty much. And know that methionine is always going to be your first amino acid because it is the start codon. Okay, so we're getting into something probably more important and definitely something that we're going to get tested on. So the idea here is that at a neutral pH, your amino group is going to take the hydrogen from the carboxyl group so that now it exists in this neutral charge called a Zwitter ion. So this whole thing is called a Zwitter ion, and the pI is the pH at which this Zwitter ion exists. So this is just saying the same thing, but know that it is the L configuration. You don't have to know these at all. Just associate L configuration with proteins. Okay. So if you have a Zwitter ion or a neutrally charged amino acid and you throw it into a base, the base is negatively charged, so it's going to want to take a proton from the amine group, right? So now the amine group is going to lose its positive charge, but the carboxyl group is going to maintain its negative charge. So now you're going to have a negatively charged amino acid known as an anode. Alternatively, if you throw it into an acidic solution, you're going to wind up with a lot of protons that are going to want to attach to the carboxyl group and remove the negative charge. So now you have a positively charged amino acid, and it's known as a cathode. So if you guys don't remember how to, memorize, anode is negative, cathode is positive. So anode has an N in it for negative, and cathode has a T, which kind of looks like a plus sign for positive. So that's how I remember it.
Phoebe, I thought anode is actually positive because when an amino acid is in a base, it's more negatively charged, and the negative charge on the amino acid is going to be attracted to the positive cathode. That's the electrical side. So it's going to be attracted to the opposite.
Yeah, because I think it's supposed to be for gel electrophoresis.
Yes, exactly.
That makes sense. I will double check that. Yeah, I'll double check that. Let's move on. Wait, does anyone else agree with that? Am I totally wrong on this?
That's what I thought when you were explaining it. I thought it was vice versa.
Yeah, I agree too.
Okay, so I'm going to double check that. I'm probably wrong on that, but yeah, I'll double check that and I'll make any changes to the PowerPoint before I post it. Sorry. Okay, so here, we know that if your pH is in a more basic solution, you're going to want to have a negative charge, right? Because you have a bunch of hydroxide ions that are going to steal the proton from the carboxyl group. And so now you're going to wind up with a negative charge, but as you become more acidic, you're going to have a lot of protons that will be able to bind to the amino group and have a positive charge to the amino acid. So a good thing to kind of remember is the pK of each of these. So the pK is going to be the pH at which the carboxyl group will be protonated. So if it's below around 2, it's going to be protonated. And if it's above 9 to 10, it's going to, the amine group is going to be deprotonated. So kind of know that the pKs for each of these groups are around 2 and around 9 to 10 for the amino group. Okay. So with glutamic acid, since it is not a neutral amino acid, it's going to have, its R group is going to play into this as well. So the R group has a different pK, but you don't have to memorize any of the pKs for the R groups because they'll give you those. So same idea here, at a very basic pH, you're going to have a negative charge, because all of the protons are going to be taken off of the carboxyl groups. And then as you become more acidic, the charge is going to be positive because now all the protons are going to be put back onto the groups. So what you will need to know about this, oh, here's a question if you guys want to attempt it. Okay, awesome. It is B. This next question, since I was wrong about the cathode and the anode, don't worry about the first question, but just try the second question. Right, everybody's saying A, and that is correct. Okay, awesome.
Wait, can you go back to the previous question? I need to write down the answer.
Oh, sorry.
Can you explain the first question for me?
Yeah, I don't understand that.
Wait, the first question?
The one that you just asked before this.
This one right here?
Yes.
Yes.
Okay, yeah. So if you have a neutral amino acid, sorry, I have to move this little tiny screen. If you have a neutral amino acid, and you know that the tryptophan is going to have a pH, when it's in a pH of 7.59, it's going to be a zwitterion, meaning it's going to be a neutral charge. If you throw that into a acidic solution, it's going to change to a more positively charged amino acid, right? Because it's going to add one of the protons or remove one of the protons, right? And then as it goes to a more basic, it's just a general question. As it goes to a more basic solution, it's going to remove the protons and it's going to have a negative charge.
So the question was asking what happens if pH decreases or increases.
Yeah, so I didn't include the pKa's of each group, but it's just a general question as to like how will it change. Sorry if it was worded really badly, but that's what I meant. Sorry. Yeah, so once it changes as the pH drops, it's going to change to a positive charge, and as the pH increases, it's going to change to a negative charge. Does that make sense?
Yes.
Okay. Yeah. Sorry if it was worded weird. Okay. So here we're going back to the Henderson-Hasselbalch equation, and knowing the ratio of conjugate base to conjugate acid, you're going to be able to kind of determine the pH of a solution. So what's more important to know is the titration curve. So know that the titration curve is going to have the pH on the y-axis and the amount of base or acid, but in this case base, equivalents that is added. So as you can imagine, as more base is added, the pH is going to increase, right? So I don't think she's going to give you a diagram on this, because I think if there would be a graph, it would be probably something about enzyme kinetics, but she's more likely to give you the pKa's, not the PI, but the pKa's, and you have to solve for the PI. So know that for a neutral amino acid, you use this formula right here. But it's different if you're using an acidic amino acid. Okay, yes, so she's going to give you all of this, and she's going to tell you glutamic acid, but she's not going to tell you if it's a acidic or a basic amino acid. You have to know that, and she's going to give you all of the pKs, and you have to use the one for the R group and the one for the carboxyl group, right?
I had a question. How would we know that it's glutamic acid versus aspartic acid or aspartate?
Wait, what do you mean by
So we know that the two acidic amino acids are glutamic acid and aspartic acid.
Right.
How would we know the difference between both of these on like a curve, or like if she asks like, oh, she wouldn't ask us like, which ones are these? Glutamic acid, aspartic acid, blah, blah, blah. You know, because how would we know the difference between them?
Right. Yeah. No, that's way too in depth. You're not going to be tested on guess the amino acid from the pKs. No, she's going to tell you like, what is the she's going to ask you what is the PI for glutamic acid, and you have to know whether to use this formula or the basic amino acid formula or the neutral amino acid formula. So you're going to have to know that glutamic acid is an acidic amino acid, and that these are the pKa's for it. But you're only supposed to use two of these, right? Like, because they're all different formulas. So yeah, she's not going to tell you to identify the amino acid on like a graph or anything like that. That's way too much into chemistry.
All right.
Yeah, don't worry about that. Okay. So for this, you would use the pK of the R group, which is this one, and the pK of this carboxyl group, which is, remember, around 2. And so obviously the PI is going to be the average of these, so it's going to kind of lie somewhere in the middle. For the basic amino acid, it's different because now you're using the pK of the amine group and the pK of the R group. So its PI is going to be higher, and it's going to be around here. So like I said, she's going to give you all three of these, and you're going to have to plug it into the formula, knowing to use your pKr and your pK2, which is the amine group pK, right? Okay. And for this, just know that titration curves are used to identify polar and nonpolar, charged and negative charge and positive charged amino acids, and yeah, it's just a general thing. And yes.
The equations, when you say she's going to give us all of the numbers, or is the graph also going to be included, or we're supposed to memorize that?
Okay, so I'm going to go back. So she will probably not give you the graph. I don't want to say no, because there's rarely ever any pictures on the exams, but sometimes they do put pictures. But she's going to give you all three of these, right? So if she was asking, what is the PI of lysine? She's going to give you all three of these, and you're going to have to know which one of the, or which two of these to pick out to plug into this particular formula.
Okay, thank you.
Did that answer your question or?
Yes, it did.
Okay. I see there's some questions here. Okay, so how would we know the amino group from the carboxyl group pK? So remember that pK for the amino group is around 9 to 10. So here we have 10.5. Oh, just kidding. 9, sorry. And then your carboxyl group is going to be around 2, so 2.2. So that's kind of what gives you the idea. So remember these like number ranges for each of those. The R group she's going to give you as pKr like this, because she's not going to expect you to memorize those. Okay. Yeah, so this is just for you to use when you do practice questions, but she's not going to expect you to memorize this. Yeah, so really just for your information, don't memorize it, because she will give you all of these values and have you solve for like PI or whatever.
So we don't need to know the R groups because I see you highlighted them.
Yeah, I highlighted them just to show you that R groups exist only for some amino acids. But like really don't worry about it because she's going to give you all of those. I think when I first learned it, I did memorize the R groups, but like when I took the test, it was kind of a waste of time because she gave you all of them.
Thank you.
So just to clarify, she will provide PK1, PK2, and the PK, the R group values.
Yes, she'll give you all three for a question, and then you have to determine what formula to use from all those three, because she's not going to be like, here's a basic amino acid, use this formula, and here are the 2 pKs that you need for it. So it's kind of like a multi-step problem.
Right. Thank you.
You're welcome. Okay, and read the summary. They're helpful. Okay, do you guys want like a 5-minute break or something before we start the next lecture?
I'm good. I can power through. Everyone else can.
Okay, it looks like the group chat kind of wants a break. So how about like 3 to 4 minutes? We'll meet back at 10:16. How about that? All right, 10:16, guys.
Primary, secondary. Pollutants. Yes. Yes. Go ahead. A question? No question. Okay. I guess let's just say hello. Hello. So anyway, so
All right, guys, is everybody back?
Yes.
Okay, thanks.
Yes.
Awesome. So I guess we'll start now. Okay, so for this lecture, it's really just an introductory lecture so you understand future lectures. So I know she throws out a bunch of like really intricate details like the hemoglobin and the myoglobin, but we're going to have an entire lecture dedicated to hemoglobin. So this is just kind of one of those lectures that you want to like look at after you learn hemoglobin, so it all comes together when you learn that after. This is just a fun little slide that is not going to be tested on. We will kind of learn about almost all of these in this course as we get through it. So don't worry about memorizing these.
Phoebe, we're still looking at the summary page.
Oh, really? Yeah, you're right. I think, oh, that's weird. Okay, that is weird. Oh, I know what I did. Is that better? I think I changed it.
Yes.
Okay, thank you so much for telling me. I'm just getting used to learning how to do this. Okay. So what I was saying is this you don't have to memorize. And for Ehlers-Danlos syndrome, we're going to go in more detail about that when we learn about collagen in a few lectures. So just kind of know kind of the general definition of it. Don't memorize this. This is just, for your information. Know that it just allows for an increase in elasticity of the skin like this guy. And don't worry about this because you're going to learn that when we go over collagen. Okay, so the main idea here is that your peptide bonds are planar. I've highlighted the planar regions, and the stuff that isn't highlighted is what is not within the plane. So kind of have an idea that it's planar. You don't have to know which ones are planar because I think that's too much detail. For these, they kind of tell you the bond strength. So remember that if you have a shorter distance between the two atoms, they have a stronger bond between the two of them, and vice versa for weaker bonds, right? So don't memorize these numbers, but kind of like look at it and be like, okay, well, the two carbons have a weaker bond than the nitrogen and the hydrogen kind of thing. Just have a general idea. Know what polypeptides are, and know that peptide bonds and amide bonds are kind of the two words for the same thing. And, you have your repeating backbone for, polypeptide chains, which is this, the nitrogen, carbon, carbon, nitrogen, carbon, carbon. Okay, so I know this is like a lot of information, and she did spend a lot of time on it. But you really, for the most part, just have to know, like, from strongest to weakest interactions, right? So your covalent bond has this much free energy that is required to break the bond, right? So you might need to memorize this number, but none of the others. If you have extra time, remember it, but I don't think it's very high yield. For these, don't worry about those. The same thing here is just you want to like look at those and kind of see the distance between the two. If it's a shorter distance like covalent bonds, you're going to have a stronger bond versus a longer distance between the two and you have a weaker bond, right? So with a disulfide bond, know that they're only between cysteines because the methionine groups has a methyl group that is going to be really hard to break, but the hydrogens are easy to remove and just, combine the two sulfurs together.
Sorry, can you just repeat what you said about disulfide bond?
Right, so disulfide bonds are only going to be found on cysteines, right? Not methionines, because methionines have a methyl group attached to the sulfur, and that's going to be really hard to break because it's a covalent bond. So it's only going to happen between the two cysteines. Okay.
Thank you.
You're welcome. So your salt bridge is kind of similar to your hydrogen bond, but the salt bridge is an actual permanent charge between the two molecules that attracts them to each other. versus a hydrogen bond kind of goes off of the electronegativity between the two ions that causes more of like a gradient of a charge, but not like a permanent charge. So that's how they are, like the oxygen is attracted to the hydrogen, right? And then, she talked about long range, but that's not going to be covered in the exam at all. It's very low yield, but just kind of know the idea that if you have two charged molecules and you put it in a polar medium, it's going to be interacting with the polar substance around it, so it's not going to have a very strong bond between the two charged molecules, versus if you throw it in a hydrophobic medium, it's, going to be able to find each other easily and have a stronger connection. And then van der Waals, which is pretty much based on dipoles, so the movement of electrons around an atom. If they all move in one direction just momentarily, then you have this like instant kind of attraction for like a second, and that's pretty much what van der Waals interactions are. As for these, don't worry about memorizing any of this. Just know kind of like strongest to weakest and a general idea as to what they are. Okay, so a reverse turn is a type of secondary structure. So it's kind of what makes it reverse in its direction. So you'll see a few of these in the secondary structure. Know that there is hydrogen bonding between the first and the fourth amino acid. So you can kind of figure it out with this formula here. So if you have number 27 amino acid, it's going to hydrogen bond with the 30th one, essentially. So for the alpha helix, this is way more important and something she's definitely going to want to test on. My TA last year, he harped on this right-handed turn. So knowing that most alpha helixes are right-handed turns, for whatever reason, I think that's important. So remember that. Because when we start talking about collagen, it gets confusing because collagen has like some other turn and whatever. So know that, and know that the R groups point outwards so they don't point towards the middle, because it's like you don't want any steric hindrance between any of the R groups if they were to all point in the middle. So it's just more favorable that way. So one of these turns, so this turn right here, you would have hydrogen bonding between these two groups. It is about 3.6 amino acids. So you want to memorize this number because I remember it being important. And know that there's hydrogen bonding between every 4 amino acids, because every 4 amino acids make one turn if you round 3.6 up. As for the actual molecular structure, the space filling structure, don't worry too much about that, but you do want to know the 3 things that I mentioned. Okay, this is something she just kind of glazed over. It's super not important. This is going to be more important when we talk about collagen, because you will see these, alpha helices kind of interacting with each other when we learn about collagen. So don't worry about this now. Just know that they can cluster together. Okay, first practice question. Awesome. So I'm seeing a lot of C's, which is the correct answer. Great. Oh, what happened here? Okay.
Hey, Phoebe.
Yes.
So what was the difference between the one that was 3 versus the one that's 4?
Oh, okay. So, going back. So it's 3.6 amino acids per turn, right? And when they turn entirely, you have, the amino acid here and the amino acid here, which are going to hydrogen bond with each other. So it's the average here, so it varies between different alpha helices, but that's pretty much taking the average, so you just round up and assume that it's every 4th amino acid that is being hydrogen bonded. So number 1 is going to hydrogen bond with number 4, and so on. Does that make sense?
Yeah, so with the I+3, that's just for like bond length, or?
Oh, I+3 is for the reverse turn, which is different. It's a
Oh,
different
okay.
type of secondary structure.
Thank you.
Yeah, you're welcome. Okay. So your beta strand, you're definitely going to want to know the difference between a strand and a sheet. So a strand is just one strand of amino acids, and then a sheet is one or more, strands. So your anti-parallel, know that it goes in opposite directions, and that one amino acid hydrogen bonds with 2 molecules of each other, right? So it's 1-to-1 essentially. And then with parallel, they go in the same direction, and these are going to be a 1-to-2 kind of interaction, right? So kind of the way I did this is I kind of drew out the structure so that I can like remember it in my head. I don't remember how she tested on it, or even if she did, but just kind of know that the difference between the two. And then she also mentioned that within, a sheet you could have both types. Okay. So this is low yield, don't worry about it. Okay. I have this question. I worded it kind of weird, but I'm hoping you guys get what I'm asking. Okay, I'm seeing like a mixed kind of thing. So the answer is A. It is a little confusing because I did kind of word it weird, but yeah, so with parallel, you're going to have one amino acid is going to attach through hydrogen bonds to 2 different amino acids on the adjacent strand, whereas with the anti-parallel, it's going to be a 1-to-1. Hopefully that makes sense. Okay. So she did talk about how you start off with a denatured protein that come out of the ribosomes, and then they all kind of spontaneously fold to produce the most energetically favorable folding shape. So you'll get the secondary structure, and then ultimately you'll get the native or tertiary structure. So remember that proteins do not have a function until they're in their native form, and usually, or a lot of proteins get in their native form when they are in their tertiary structure. However, other proteins like hemoglobin need to be in their quaternary structure for them to have a function. I think somebody's mic is on. Do you have a question? Okay. So yeah, she starts to introduce myoglobin, and don't worry about this now because we're going to get into it in greater detail, but pretty much this is your heme group, and it's got like an iron in the center, and then this is your protein that surrounds the heme group, and it's similar to hemoglobin, but hemoglobin has 4 of these whole, structures put together in a quaternary structure. The number one thing that you do need to know about this slide is this thing that's highlighted in blue. You have to know that the primary structure is what dictates the tertiary structure, right? So your sequence of amino acids is going to determine how it folds, because if you have a bunch of hydrophobic amino acids, they're going to want to be towards the inside, and then the hydrophilic ones are going to want to be towards the outside. So that's going to change the way it forms. So I think there was a question about this on the exam, so make sure you know it, and know that the tertiary structure is the 3-dimensional structure. And here she was just kind of giving you an example of these, but don't worry about it. Yes, low yield. Okay, so the main point here is what I just said, the fact that hydrophilic amino acids are going to be on the exterior and more hydrophobic amino acids are going to be on the interior. And that's just because if these proteins are just floating along in your cells, that's, your cells are made of water, so they're going to want to, have the hydrophobic amino acids hide out in the center to minimize its, touching of the water. But that's not the case for all types of proteins. So you have an aquaporin, like you see in many types of cells. You have your plasma membrane. So this goes through the plasma membrane and allows water to enter the cell or exit the cell, whatever. So your plasma membrane, it's got your phospholipid tails, which are hydrophobic. So you're going to want your hydrophobic amino acids on the exterior and your hydrophilic amino acids on the interior to allow water to move through. So this is just kind of a summary slide. I've kind of written out the main ideas for all 4 of these. So just kind of know this, use it to test yourself on the main ideas. But once we start learning about more types of specific proteins like hemoglobin or collagen, it's going to make a lot more sense. Okay, so this is a question. Go ahead and attempt it. Okay, so it's kind of a mixture of a bunch of different answers. So the correct answer is F. It's kind of a trick question. Remember that your primary structure is essentially your polypeptide sequence, right? So it's the amino acids all linked together making up a polypeptide, and that sequence is your primary structure. And your primary structure, along with that sequence, are what dictate the tertiary structure.
So the terms primary structure and polypeptide sequence can be used interchangeably?
So essentially, yes. I mean, polypeptide sequence is just a way of describing a sequence of amino acids, right? So you've got cysteine, methionine, glutamic acid. That sequence is a polypeptide sequence. And then that makes up your primary structure. Does that make sense?
Yes, thank you.
You're welcome. Okay, this is a transition slide. Okay, so here she wanted to give you an example of quaternary structure. So quaternary structure is going to be more than one proteins put together through the same types of bonds that you would see in tertiary structure. So you know how your primary structure is made up of covalent bonds or peptide bonds. Your secondary structure is going to be made up of hydrogen bonds, and then your tertiary and quaternary structure are going to be made of all of those other bonds that we talked about at the beginning. So it could be, 2 or more proteins coming together and interacting with one another. So she's given you an example of 2 proteins that come together, and then, a tetramer here. So this is hemoglobin, essentially. But you don't have to worry about hemoglobin yet. So I would kind of have an idea as to the difference between these. So a homodimer are 2 proteins that come together that are exactly alike, right? A heterodimer are 2 proteins that come together that are different. A heterotetramer is a total of 4 proteins that are different. So it doesn't matter if all 4 of these are a different type of protein. As long as there's 2 different types, then it's considered hetero, right? So this is kind of the example that I'm trying to get at. So a heteropentamer, you see that there is 2 of the same type with 3 of a different type. It's still going to be hetero because it's not like this where it's all the same type. So have an idea as to what these mean. I don't remember if there was a question on this, but I vaguely remember having to memorize this. And these are just examples. Don't worry too much about these. They're way too much detail. Same here. Like, you can look at this and just, kind of have an idea as to different types of quaternary structure, but she's not really going to test you on it. Okay, so this is a little bit more important. So here you see a bunch of cysteines engaging in disulfide bonds. She said that an oxidative environment would be good for this because the oxidative environment would allow for the removal of the hydrogen so that these two can bind together, right? So when you want to break these bonds, you would use beta-mercaptoethanol. So remember this name. This is important to remember. Know that it is a reducing agent because it allows for the breaking of these sulfide bonds to disulfide bonds to allow for them to go back to their normal state with the hydrogens attached to them. Okay, so I think I also remembered this in order to denature a protein. So you have your tertiary structure here, and then once you break the disulfide bonds and you add urea, which also helps to denature the protein, you're back to your primary structure, which is literally just the sequence of amino acids all linked together by covalent bonds, because covalent bonds are really hard to break. So you can't just break them with beta-mercaptoethanol. They actually need to be hydrolyzed, which we'll get into. So remember this, and remember beta-mercaptoethanol especially being useful for breaking the disulfide bonds. Okay, so for this, she's not going to show you this like graph on the test or anything like that, but it is important to know that tryptophan and tyrosine will absorb at 280 with UV spec. And she also mentioned that phenylalanine absorbs around 260, but she just mentioned that as like a side, comment. So don't worry too much about memorizing that. But I think there was a question where you had to determine the identity of an amino acid based on her giving you one of these as like a clue. So know that. She said not to worry too much about this slide because she does go into the Edmond degradation in the next slide, but I would take away that a strong acid can break down peptide bonds, right? So if you have HCl, for example, that would hydrolyze the peptide bond and allow for the release of free amino acids in an attempt to sequence a peptide if you wanted to do that. But she's not going to test you on that. It's just more of a fun fact. Okay, so for the Edmond degradation, pretty much what she's going to want you to know is that the Edmond degradation will allow for sequencing of a peptide by removing the N-terminal amino acid. So you'll have this substance, this substance that will bind to, I think somebody's mic is on. Okay, so your phenyl isothiocyanate will bind to the N-terminal amino acid and just remove that N-terminal amino acid. So you would have to do multiple rounds to break down the polypeptide one by one. So that's pretty much all you have to know for that. You will have to memorize these proteases and their specificity, and also know that they cut at the carboxyl end, right? So they're not going to cleave at the amino end. So know that because you don't want to get confused on the test. I know she asked the question asking you to determine a polypeptide sequence based on, the findings of using trypsin or chymotrypsin, etc. So she provides a practice question at the end of her slide. So make sure to do that because it will be a question on the exam. So know that it always cleaves the carboxyl end, not the amino end. And don't get that confused with Edmond degradation, which takes the N-terminal instead of the C-terminal. Okay. So luckily for us, we don't have to know how mass spec works. You just have to associate the word primary structure and mass spec with each other. So if she asks you, how would you determine primary structure, and mass spectroscopy is one of the answer choices. sorry, there's a question in the chat. Did she say not to know bromelain? So I remember that being low yield, but honestly, I would remember it anyway, just in case. But if you don't have the time for it, then don't worry about it, because if anything, she's going to test on these two.
She said yesterday we didn't have to know bromelain.
Mm-hmm.
Okay, then don't know
Well, that's
bromelain.
good,
I think
dude.
I
I'm
memorized
glad.
it anyways,
So
but
she like
yeah,
is
good
it
to know.
just logos, or does she build websites too? I can't remember.
Okay, so yeah, so she's gonna ask, how do you find primary structure? Mass spec is gonna be an option, and you're gonna choose mass spec. Same thing with secondary structure, just associate secondary structure with CD spec, and tertiary structure with X-ray crystallography. That's really as easy as it gets. So last practice question.
Gotcha. Oh man, dude, yeah, you are never gonna find him.
Oh my god, did I change the question? Wait, I put tertiary structure here, and I changed it to secondary structure. Hold on, guys. So tertiary structure is X-ray crystallography. Yes, sorry, I changed the question in the answer. So sorry about that, but yes, A is correct.
That's
and then secondary structure, it would be CD
crazy
spec.
because that's the difference between him getting
Great.
like
So
he
read
could
the
have
summary,
like
and she
multiplied his
has these
business by like
practice
7 or
problems.
8-fold.
So make sure to
by
do those
being on
because
the
they're going to
front
be pretty
page.
similar to what you'll see on the test. And same with the fill in the blank, they're also helpful. I will post my answers to these because she's not going to post the answers, I don't think.
Yeah, right.
So I'll
It would
post
be a
my
fair
answers to
price,
these sometime
yeah.
next week to give you guys time to attempt them.
That's crazy, man. Good for her.
Yeah.
That is such a good
So
marketable skill.
that was the end of that. Does anyone have any questions about either of those two or any other general questions?
She's already posted the answers to her questions and fill in the blanks under her modules.
Oh, awesome.
Yeah, dude, you love that.
Cool. Good to know. All right, so make sure to attempt those before you actually go and look at the answers because they're super helpful.
Oh, yeah.
Does anyone else have any questions? If not, you guys are free to leave.
Phoebe, just getting back to the first PowerPoint where we ran into the hiccup with the anodes and cathodes. I was thinking about it. It can't either of them be positive or negative depending on the cell solution you're in. I know it has to do with redox reactions. I can't remember off the top of my head.
Right, yeah. So I understood that slide as, let me go back to it.
Right.
That's the oil rig in your second presentation, or your second PowerPoint, and that reminded me about that.
Yeah, so I understood it as you dump the amino acid in a basic solution, so it's going to be negatively charged, right?
Mm-hmm.
So that's how I understood it. Is that what you're trying to say?
I'm not necessarily trying to say one way or the other. I was just maybe seeing if that would spark something in your memory. I can't tell, honestly. I can't remember. I have to read up on that again.
Yeah, obviously I do too. So
someone had written in the chat that in electrophoresis, the anode is the positive pole and the cathode is the negative pole. And that is the case. And so then that means that the way that you wrote it on your slides would be correct. Because if you put the amino acid in the basic solution, it would become positively charged and it would go towards the cathode, which is the negative pole.
Yeah, man. That's
and
cool.
the vice versa. If you put the amino acid in the acidic solution, it would become negatively charged and it would be attracted to the anode, which is the positive pole in electrophoresis.
I think if you switch that around, like when you're in an acidic solution, that means there's excess hydrogens, so you're going to become more positive. And when you're in a basic solution, there's a lot more hydroxyls, so it's going to take the hydrogen, so you're going to become more negative.
Right, yes, that makes sense. So wait, what did that have to do with?
So I think they can be both. It depends on the solution, doesn't it?
Right, yeah, it depends on the solution that it's in.
So connected, man. All that stuff seems so bureaucratic when you view it from the outside, but when you view it from the inside, you're like, oh no.
So should we really memorize anode and cathode as positive and negative, or vice versa?
People like have to come up with all of these really complicated laws.
I will get back to you on that, but if I remember it being very low yield because it's too much chemistry and not enough of like the big picture stuff that she wants us to know. But I will email her and get back to you.
Yeah.
Maybe I'll make a Canvas announcement or something, and we could clarify that.
Okay. All right. Thank you so much for your time, Phoebe. You were great. I really appreciate it.
Yeah, thank you. I'm happy I was helpful to you guys.
Yeah. I know,
so yeah, like I said, if you guys have no questions, you're free to leave, but I'm going to stay around a little bit longer to answer any questions you guys have.
Yeah, man. I got in a wreck, not like a wreck, but
Thank
I got
You're welcome.
fender-bendered not too long ago, and I didn't have to pay anything though, which was nice because
I think that I'll just clarify
it sucks, though,
when the pH is decreased, so they mean that you put the amino acid in an acidic solution, the carboxyl group becomes protonated, so it moves from being a Zwitter ion to being a positively charged amino acid, and that's why it moves towards the cathode, because the cathode is negatively charged in electrophoresis.
Yes, exactly.
That makes total sense. Yes.
Yeah, so that means that what you wrote, Phoebe, on your slides is correct.
Okay, awesome. That is great to know.
I got in here and like the difficulty of the coursework and material of biochemistry, genetics, anatomy, microbiome, and I'm like at like a med school level, like we're using.
Yes. Oh, yes. Go ahead. Sorry.
The bicarbonate system, the buffer system.
Mm-hmm.
The example that you gave us, we use instead of the when the H plus increase, the reaction will shift to the left, right?
Correct. Yes.
And then how about, can we use it also when carbon dioxide increase?
So if carbon dioxide increases, it's going to cause the reaction to make, or cause it to make more carbonic acid. And that carbonic acid is going to want to dissociate into protons and bicarbonate. So it's going to increase the acidity of the blood. Does that answer your question?
You have to write it down. So we cannot use for carbon dioxide. We have to use it only for when the H plus is increased. Like I'm trying to think, like if I have a lot of carbon dioxide in the blood, so can it be applied to the buffer system or no? Like it's going to shift to the left or to the right, or to stay the same?
Right, so that was kind of what we talked about at first. Here, I'll draw it out for you on this slide. Can you see it okay?
Yes, thank you.
Yes, okay. So if you increase your carbon dioxide, it's going to shift to the right, and it's going to make more bicarb, and then as a result, the bicarb is going to dissociate and make more protons, and more bicarb as well. Carbonic acid is going to dissociate into protons and bicarb, right? So it's going to shift to the right. Does that make sense?
Yes, thank you.
You're welcome. I saw a couple questions in the chat. So if we come up with any questions later, is the best way to reach you through email? Yes, please email me. I have it linked to my phone, so I'll see your emails right away. Like I said, I posted it on the first slide, at the intro slide. So hopefully you guys have that. So the buffer system, what about it did you want to go over again?
Oh, you already answered the question. Thank you.
Okay, great. Awesome.
Could you explain KEQ one more time?
Yeah. I'll just put up that slide.
Thank you.
Yeah, no problem. Okay. Let me know if you guys can see it. Okay, so KEQ is the concentration, or I'm sorry, the equilibrium constant, right? So it's going to be equal to the concentration of the products, which are these two, and that's the dissociation of the weak acid. So
we can only see the first slide.
Like I'm sorry. Let me try. Did it change or no?
No.
Oh my gosh. Sorry, I'm still trying to figure out how to use Teams.
We're seeing your presentation slide.
Oh, the first slide?
Mm-hmm.
Yes.
Okay. Let me see if I can fix that. Um, did it change?
Yes, now we can see slide 3.
Okay, awesome. Okay, sorry about that. So for the equilibrium constant, it's going to be equal to the concentrations of the protons and the conjugate base that happens when the weak acid dissociates, right? So that's going to be essentially the product over the concentration of the non-dissociated weak acid. So this is essentially giving you a ratio of how much it dissociates, right? So if it dissociates more, you're going to have more protons in the medium. And when you have more protons in the medium, you're going to have a drop in the pH. So it's going to be more acidic. So that's why when your products are higher than your non-dissociated weak acid, it's going to be a large KEQ because this number would be in the numerator here. And that's how you know it's going to be a stronger acid because there's going to be more protons. I think this is flipped when you take the negative log of KEQ because pK is still kind of giving you an idea as to how acidic something is, but it's using like easier numbers. So with pK, it's going to be the opposite. So a lower number is going to be more a stronger acid as opposed to a larger number like you would see in the equilibrium constant. Does that answer your question, or is there something more specific you wanted to go over?
No, that's good. Thank you.
Okay, awesome. Are there any other questions?
Do you mind if you go a little over again about the titration curve, and which formula to use when she asks us those questions, please?
Yeah, of course. I'll have to switch over to the other slide, so I may have the same problem as before, but let me know if you can see it or not. Is it showing or?
Not yet.
Okay. Is it showing yet or no?
Yes, now the first slides.
Okay, awesome. So you wanted to go over titration curves again. Oh my gosh, I changed it to the wrong thing. Sorry.
It's okay.
Okay, so you wanted to go over titration curves. Okay, so for example, if she were to say use glutamic acid in this example because it's right here. So she would give you these 3 values. So your pK1, since it's 2.2, you know that's going to belong to the carboxyl group, right? Your pK2, on the other hand, is going to belong to your amine group because it's around 9 to 10. Then she'll give you your R group, which she will give you the R. So you know that that's the side chain. So she'll get these 3 numbers and she'll be like, oh, what is the PI of glutamic acid? So the first step that you need to know is that glutamic acid is an acidic amino acid, right? So then you have to look at all of these and think, okay, I need to use this formula for an acidic amino acid to find the PI of it. So you would take the R group pK, so 4.3 in this example, so 4.3, and then you would add it to the carboxyl group, which is going to be 2.2. Right? And then you add those two and divide by 2, and that gives you the PI. Does that make sense?
Yes, thank you.
You're welcome.
Hi, Phoebe. I had a question.
Yeah, go ahead.
For slide 23 for the titration slopes, I was writing something about steeper. Can you go over the take-home message on that one slide?
Yeah, sure. You said it was 23?
Yeah. I think on the intro.
Is it this one?
Yeah.
Okay. Yes. So take home message is if you have a steeper slope here, you're going to have a better buffer because it only, like for example, this one, you see that it only changes by like 3 pH amounts, right?
Mm-hmm.
And even though the ratio of conjugate to base changes dramatically, right?
Mm-hmm.
So if you had like a slope, like a buffer that had the slope of this, you'll see that it changes way more than this little triangle does. And you're going to have a larger change in the pH, but with less of a change in the ratios of acid to bases. So, so a steeper slope kind of indicates that it's a better buffer. Does that make sense?
Yeah, it does make sense. And then for like a test question, would she just give us a range of buffers and tell us which one would be better? Because I see you have the phosphate buffer, the amino, the So between those 3.
If she did give you a question on this, she probably won't give you like a graph. I'm just kind of throwing that out there in case she does, because I don't want to be like assuming that, she's automatically not going to do that. But if she did, she will probably just, kind of give out if the base is equal to the acid, know that the pK is equal to the pH. And I don't know, I can't really think of a question offhand off of that, but just kind of understand like this basic concept that the best buffering range is right here where the pH equals the pK, because you have this whole area of it's right in the middle of this whole thing, so it can go either, it can drop easily and it can increase easily, right? Because it's in the middle.
Okay, perfect. Thank you so much. I was so confused with like looking at all the numbers. I was like, okay, so we just need to know like the pH when it equals pK basically.
Yeah, so that's your best buffering range. Like what I highlighted pretty much is like the take home message.
Perfect. Thank you so much, Phoebe.
You're welcome. Okay, does anyone else have any questions? If not, you're totally free to leave.
Hi, Phoebe. Are you going to post these with the notes for us, please?
Yes, I will post them later tonight after my PM session. So sometime around 7 to 8 p.m. tonight, if that's all right.
Okay, thank you. So you will come back at 5 p.m. again tonight?
No. So the 5 p.m. lecture is the same exact thing that we just did right now. It's just at 5 p.m. for those people who don't like waking up early or have something else going on in the mornings.
Oh, great. Thank you. Perfect.
Yeah, you're welcome. All right, if there's no more questions, I'm going to go ahead and end the meeting, and I'll see you guys next week or tomorrow if you're going to study skills.
so I had the exact opposite experience where my parents and my grandparents my mom isn't super politically charged, but my dad and my grandparents are super politically charged. And I had like this